Yes, we can delete memories, but the science and the fiction of memory manipulation. Now, the first thing we need you to do, Mr. Barish, is to go home and collect everything you own that has some association with Clementine. Anything. We'll use these items to create a map of Clementine in your brain. So we'll need photos, clothing, gifts, books she may have bought you, CDs you may have bought together, journal entries. We want to empty your home. We want to empty your life of Clementine. After the mapping is done, our technicians will do the erasing. This sounds like science fiction, and of course it is. It's from the film Eternal Sunshine of the Spotless Mind. But the questions it raises about memory, about whether we could actually erase memories, those are very real. Last week, we explored how memory works, how our brains encode, store, and retrieve different types of memories. Our brain isn't like a computer hard drive, where files are neatly stored and deleted. Instead, memories are reconstructed each time we remember them, with many brain regions working together to assemble the created experience. This week, we'll use Eternal Sunshine as a lens to explore whether we could manipulate memories in a similar way to how Joel has his memories manipulated in the film. To find out, let's first review the plot, and then we'll ask 3 questions. 1. Is it possible to prevent memories from forming? 2. Can we erase them once they've formed? And 3. Could we implant entirely new memories that never happened? Before we begin, if you haven't seen Eternal Sunshine of the Spotless Mind yet, you really should. This is one of those movies that's better experienced without knowing too much about it. And if you have seen it already, you'll know that it's a fantastic movie to rewatch. What is the plot? The film follows the relationship between Joel, played by Jim Carrey, and Clementine, played by Kate Winslet. But the story is told in a deliberately non-linear way. The film begins with what seems to be a chance meeting between 2 strangers on a train to Montauk. But something feels off, awkward even, and there are some strange gaps. Joel is confused by the name Clementine. It's as if he's never heard it before. This opening scene is not the beginning of the story. It's not the first time Joel and Clementine have met. After the opening scene, the timeline shifts and we learn that Joel and Clementine have already been in a relationship, and it ended badly. Joel has discovered that his now ex-girlfriend Clementine has undergone a procedure at Lacuna Incorporated to erase all of her memories of their relationship. Devastated, Joel decides to undergo the same procedure. As the technicians systematically erase his memories, Joel begins to experience these memories one last time, starting with their bitter end and working backwards through their relationship. He experiences the fights, the joys, and the tender moments again, and he realizes, possibly too late, that he doesn't actually want to lose these memories after all. The film's premise taps into something deeply universal about the human experience of pain and memory. Who hasn't wished they could erase a painful memory? The devastating heartbreak of a failed relationship, the grief of losing a loved one, the shame of an embarrassing moment that kept you awake at night. These deeply human desires to escape our painful memories makes Eternal Sunshine's premise so compelling. Not just as science fiction, but as a mirror to our own longings and fears about our memories. But is such precise memory manipulation actually possible? Question 1. Could we prevent memories from forming? The year is 1967. A goldfish swims lazily in a 2-sided tank. As Mr. Goldfish is exploring the south end of his tank, the water around him suddenly brightens. He continues swimming unbothered by the light. But 20 seconds later, zap, a mild electric shock jolts through the water. The shock doesn't hurt, but it's not exactly pleasant either. So he darts over the barrier into the darker north side of the tank. The peace, unfortunately, doesn't last long for Mr. Goldfish. The north end of the tank is soon lit up. And again, zap. So back to the south side he goes. Over the next 40 minutes, Mr. Goldfish learns something important. Every time the light comes on, he should swim to the other side of the tank to avoid being shocked. Mr. Goldfish is one of dozens of goldfish swimming in tanks at a University of Michigan lab. Little do these fish know they're about to help scientists answer a mysterious question about how our memories work. When do experiences become permanent memories? And could we prevent these memories from forming? Before 1967, scientists had a hunch that whether an experience turned into a long-term memory or not had a lot to do with proteins. Inside every cell in our brain, including neurons, we have tiny molecular machines called ribosomes. These ribosomes work a little bit like an assembly line, linking together amino acids to form proteins. This process is called protein synthesis. Proteins are essential for neurons. They allow neurons to communicate and form new connections with other neurons. No proteins, and neurons can't do what they usually do. The scientists wondered, if protein synthesis is crucial for neurons to form new connections, what would happen if they blocked protein synthesis right after learning? Without the ability to make new proteins, neurons couldn't communicate or create new connections. Would this prevent memories from forming altogether? To test this idea, the scientists at the University of Michigan used an antibiotic called purimycin. Purimycin stops ribosomes from making proteins. The researchers had a simple but very clever plan. Teach the goldfish a lesson, then use purimycin to block protein synthesis at different times after learning. A third of the goldfish were injected with purimycin immediately after training. Another third were injected 30 minutes after training, and the final third an hour after training. When tested 3 days later, the results were striking. The goldfish that received purimycin immediately after training did not swim to the safe side of the tank when the light came on. They had forgotten what they had learned. It was as if the training had never happened. The fish that received the injection an hour later, they remembered perfectly well, and those that were injected at the 30-minute mark showed partial memory loss. What the scientists had discovered was a crucial window. Somewhere between 0 and 60 minutes after learning, memories are consolidated, at least in goldfish. The timing was crucial. Block protein synthesis immediately after learning, and memories never form. Wait an hour, and the memory is fully consolidated, safe from the effects of purimycin. For decades after this discovery, most scientists believed that memories were fragile while being learned, but became permanent once consolidated. It would take another 33 years and a curious neuroscientist named Kareem Nader to challenge everything we thought we knew about memory storage. Question 2. Can we erase memories once formed? In 2000, a young scientist named Kareem Nader, who had recently started working in Joseph Ledoux's lab at New York University, walked into Ledoux's office with what seemed like an absurd question. Could we erase an existing memory? The idea seemed so ridiculous, Ledoux's reply was, "There's no way that would work." Remember, at this time, scientists thought memories were more or less permanent once they were consolidated. But Kareem had a hunch. What if memories became fragile again when we retrieve them? Against Ledoux's advice, Kareem decided to run the experiment anyhow. secretly. First, he taught rats to fear a tone by pairing it with a mild foot shock. A rat's response to fear is to freeze. So when the rats started freezing when he played the tone, Karim knew they had learned the tone meant a shock was imminent. He waited 2 months to ensure the memory was well consolidated. Then came the crucial test. Karim played the tone, the rats displayed their freezing response, and then he injected anisomycin directly into their amygdala, the brain area known for the role in fear responses. Like puromycin, anisomycin works to block cells from making proteins. The next day, when Karim played the tone, the rats did not freeze. They showed no fear of the tone. It was as if the memory had been erased. To show this wasn't just a general brain disruption, Karim taught rats to fear 2 different tones. Then, during the erasing procedure, he gave the anisomycin when just one of those tones was played. The next day, the rats had only forgotten their fear of the targeted tone. The other memory remained intact. Karim bounced back into Ledoux's office with the news. It actually worked. The finding was revolutionary. It suggested that the mere act of remembering makes memories vulnerable again. It provided more evidence that when we recall a memory, we aren't simply reading it out like a computer file. We're reconstructing it, and in doing so, we open a window where that memory can be modified or erased. Imagine what this might mean for millions who suffer from PTSD. Soldiers returning from war, survivors of assault, first responders haunted by tragic scenes. Traumatic memories aren't simply harmless afterthoughts. For so many, painful memories trap people in a cycle of fear, anxiety, and depression. While anisomycin used in the rat studies is too toxic for humans to use, researchers have found other drugs that might have similar effects in humans. Early clinical trials suggest that giving patients these drugs while they remember traumatic memories can help reduce their emotional impact. But it's important to understand what's happening with this type of treatment. This isn't like the complete memory erasure depicted in Eternal Sunshine of the Spotless Mind. In Karim's experiments, anisomycin was injected specifically into the amygdala, a brain region critical for fear responses. What was likely being disrupted was not the entire memory of the event, but rather something more specific. The rats likely still retained the memory of hearing the tone and may have even remembered the experience of being in the chamber. But the fear response connected to that memory was erased. We'll come back to this point later on. Question 3. Could we implant entirely new memories that never happened? Let's travel back to the 1960s, when researchers at UCLA were making an extraordinary claim, one that seemed almost impossible to believe. While scientists at the University of Michigan were discovering ways to block memory from forming in goldfish, these UCLA researchers proposed something even more radical. They believed they could transfer memories from one animal to another. Their method was surprisingly straightforward. They extracted RNA, a type of molecular cousin to DNA, from rats that had developed a learned fear response. They then injected that RNA into untrained rats. The astonishing claim was that the untrained rats appeared to inherit the learned behaviors from the trained rats. This wasn't just an isolated finding from one lab either. Other research groups began reporting similar results, suggesting they too could transfer memories through RNA. But as the saying goes, extraordinary claims require extraordinary evidence. And some scientists were simply unconvinced that the evidence was extraordinary enough. When they attempted to replicate these studies using more rigorous experimental controls, they failed to reproduce the original results. A scientific consensus formed. There's no compelling evidence that memories can be transferred between animals. In fact, for decades afterwards, making claims about memory transfer became somewhat of a taboo topic in neuroscience. There has been some re-emergence of this topic, but it still remains controversial. While these early experiments on memory transfer failed to stand up to scientific scrutiny, they did raise an intriguing question. Could we artificially create new memories that never actually happened? Half a century later, scientists finally had tools sophisticated enough to tackle this question. A revolutionary new method called optogenetics was making waves in the neuroscience community. Optogenetics is kind of like having a remote control for the brain. To set it up, scientists use a genetic technique to add special light-sensitive proteins to specific neurons in a mouse's brain. Once these proteins are in place, the researchers can switch these neurons on and off simply by shining a light onto them, just like flipping a light switch. The trick is determining which specific neurons to target. In one study, the researchers knew that when a mouse forms a memory, only a subset of the neurons are active. So they designed their system to add the light-sensitive proteins only to neurons that were active when the mouse was in a particular box. Let's call it box A. Box A had distinct features, specific visual patterns on the walls, a unique smell, a particular texture on the floor. These are all things a mouse would use to recognize its environment. The next day, they put the mouse in a completely different box, box B. Here, they did two things simultaneously. They gave the mouse mild foot shocks while using light to switch on those box A neurons. In other words, they artificially activated the neurons that had been active while the mouse was in box A. The researchers reasoned that by reactivating these neurons, they might make the mouse remember or even experience what they experienced while in box A, but while they were actually in box B receiving the foot shocks. The results were striking. The next time the mouse was placed in box A, it froze in fear, even though nothing bad had ever happened in box A. By activating the box A memory neurons during the fear experience in box B, the researchers had created a false memory. The mouse now seemed to remember being shocked in box A, even though it had never occurred. Timing of the light activation was crucial. False memories were only implanted when the neurons were activated during the shocks. If they activated the neurons before or after the shocks, then no false memory was formed. While these results are remarkable, it is important to understand that there is a significant gap between learning that a tone means a shock is about to happen and the complex autobiographical memories we see manipulated in science fiction. So, how far are we from turning science fiction into science fact? In real life, scientists can create a simple associative memory, linking a particular context with a fear response, but they can't implant specific complex memories that involve multiple sensations, emotions, and narratives, like memories of conversations or relationships or life events. Fear conditioning in animals involves relatively straightforward neural circuits, primarily in the amygdala, and this sort of memory is thought to be largely the unconscious type. But human autobiographical memories, like Joel's memories of Clementine, don't exist in isolation. We can't just go in and erase Clementine from Joel's brain as if there were a Clementine part that we could easily access. The neurons involved in Joel's memory of Clementine are distributed across Joel's brain. They interweave sensory details, emotions, semantic knowledge, and temporal sequences in ways we're only beginning to understand. But to complicate matters even more, the neurons involved in Joel's memories of Clementine are likely active for other memories too. For example, some neurons that fire when Joel remembers the sound of Clementine's voice might also fire when he remembers his mother's voice. So, while it is remarkable that science can block, modify, erase, and even artificially create memories, we are still far from the kind of precise, selective memory manipulation depicted in sci fi films and books. But that doesn't make movies like Eternal Sunshine any less fun to watch. Next week, in this week's and last week's articles, we've seen how malleable our memories can be. All this talk of the fragility of memory might have us wondering how much of what we remember actually happened. Next week, let's explore the fascinating and sometimes disturbing science of false memories. The implications are huge. Could our sense of self, our relationships, and even our life decisions be shaped by memories that never actually happened? Thank you so much for listening to this article. I just want to take a small moment to say a really big thank you to all of you. Thanks for listening to this article and engaging with the content. It really does mean the world to me. If you happen to be listening to this on a platform other than Substack and you would like to join a discussion about this content, you can find a fantastic community over at susietravis.substack.com. I'd love for you to be a part of it. It's free to join, so why not give it a try? And to all my friends who are already part of the Substack crew, I just wanted to say thank you so much. I appreciate you so much. Reading your thoughts and feedback is such a gift, and I'm so grateful for your support. So thank you so much.